All right, thank you all again. And I want to start by having us all give a huge round of applause to all the people who contributed posters, presentations, lightning talks, all the parts that made the community presentation. It's incredible, the breadth. I don't know if any of you were like me, but I tried to get around to everything. I fell short of my goals, and I'm very hopeful that at the end of this, George will tell me everything's on the internet so that I can be sure to view all the posters and things that I might have not been able to get to. But once again, absolutely tremendous. And just as a reminder, you've just received an email with a link to please go to and vote on your favorite presentations, your favorite lightning talks, and so forth, which will be presented at the end. So it's my great honor to be up here today with two of our collaborators, partners from the FDA, Azadeh Shoaibi and Alan Williams from FDA's Center for Biologics Evaluation and Research. They're going to be talking about a program called BEST, and I will let them describe it in further detail, but I just wanted to note that BEST, as a collaboration between FDA and the OHDSI community, represents a truly significant step forward for OHDSI and its mission. The collaboration partners thus far who've had the opportunity to engage in BEST include Stanford, Regan Streif, UCLA, Columbia, Georgia Tech, IQVIA, with opportunities for many more in the future. It is a partnership between FDA and OHDSI and others, and you'll hear about that as Azadeh and Alan talk. In the context of their presentation, I'd like to just harken back to what George and Patrick referenced, which was the mission of OHDSI that was stated at the beginning, which was to generate the evidence critical to decision-making to improve health. And OHDSI is not just about evidence generation, even though I'm very happy to have 18,000 new papers that I'm going to be putting my name on, thanks to Martijn. But it's about the delivery of that evidence into care. And when that mission was created, OHDSI was not having 500-person parties and breakfast burritos and all of that. It was a group of people, many of whom are in this room today, who had recognized that the expertise and the data that they had was important and could be valuable, but the passion and the commitment to making a change and driving evidence to improve decisions was what that mission needed to be. And so it's so exciting to be here and to be able to be up on this stage as Azadeh and Alan talk about BEST, which is really a true step forward in that direction. So let me turn things over to Azadeh. Thank you.
Thank you, John. Good afternoon, everyone. My task is to talk about the BEST initiative, which is a new program under the larger umbrella of the CBER Sentinel program. So first, I'm going to describe what biologics are, since some of you may not be familiar necessarily with biologics, and then introduction of the program and where are we planning to go from here. CBER regulates a diverse array of medical products, and we call them biologics. They include blood products and blood components, vaccines, human tissues and cellular therapies, and a large array of other products. And the surveillance activities that we conduct under the CBER Sentinel program assess the safety and effectiveness of these medical products. CBER has been conducting active surveillance for many years with many different organizations through either agreements or contracts. One of those activities that probably most of you are familiar with is under the umbrella of the Sentinel program, a contract with Harvard Pilgrim Healthcare Institute. But we have also had a long history of working with other organizations such as IBM or Health Corps, and some federal agencies, sister agencies such as DOD or CMS, etc. So with the kind of long history of active surveillance work on mainly claims data, we gradually at CBER came to the realization that biologics have certain characteristics that would require certain other characteristics to be built into the infrastructure for surveillance activities. So in order to accommodate those certain characteristics, CBER decided to invest in some additional programs that, again, fall under the larger umbrella of the Sentinel program. And we attempted to accommodate these unique characteristics of biologics to expand and scale up our surveillance capability and capacity, and also utilize more advanced technology and methodology that are available. So as a result of this, CBER awarded 2 one-year contracts in September of 2017 to OHDSI and IQVIA working together, and we called it BEST, Biologics Effectiveness and Safety Initiative, and we started it as a one-year pilot program. So the BEST initiative consists of 2 contracts, and for just simplicity, we call them Contract 1 and Contract 2. Contract 1 mainly focuses on building infrastructure to develop advanced and large-scale surveillance capabilities to meet the biologics needs in terms of data and analytics. And Contract 2 focuses on more methodology and innovative methods to use these methods such as machine learning and artificial intelligence and others to mine unstructured text from EHR data sources to advance our surveillance capabilities. So Alan will be talking about Contract 2, and I will be focusing on Contract 1. As John mentioned, we have several collaborators from the OHDSI community, including UCLA, Georgia Tech, Reagan Street Institute, Stanford, and Columbia Universities, and IQVIA serves as the coordinating center for the BEST initiative. So we have access now both to claims and EHR data sources, and we use the OMOP Common Data Model. IQVIA and Reagan Street provide about 100 million patient records in the form of claims data, and Stanford and Columbia Universities, along with Reagan Street Institute, provide us with EHR data for approximately 24 million patient records. So what did we accomplish in the past year? Because the contract is actually the 2 one-year contracts are ending this month. So the focus of this one-year pilot program was to build some new infrastructure. And that was the main focus of our work. And also we focused in terms of products on blood components. And the reason for focusing on blood components was that we had a history of having worked with claims data, and claims data have major limitations for identification of blood transfusions. And also, in the past few years, a new coding system by the name of ISBT 128 coding system has become available, which is now the standard for identification of blood transfusion. ISBT stands for International Standard for Blood and Transplant, and it provides comprehensive and granular information about the number of units of blood, component that is transfused, as well as specific attributes of the blood component. So availability of the ISBT-128 was also very crucial for now that we had access to EHR data to be able to focus more on blood components. Because the OMOP Common Data Model has a certain level of flexibility and expandability, we were able to incorporate the ISBT coding system into the Common Data Model and quickly start to use it across the sites that provided the EHR. As part of building the infrastructure, we also reduced the data lag that we previously had working with other contracts, which was about a year. So we reduced it to about a few months. And also, we now have access to analytics that are applicable to diverse and large-scale data sources so that we can scale up our capacity. So one of the projects that I would like to present here as an example of what we have worked on is we looked at the utilization of blood components across the 3 sites that provided the EHR data to us. And we compared the capturing of blood component transfusion using both ISBT codes and billing codes. So here, on this very busy and small diagram, what I'm showing is we looked at the number of patients who received some kind of blood component transfused. And we had 3 sites. We looked at red blood cells, platelets, and plasma. The blue lines here show the capture of blood component transfusion using ISBT codes, and the orange lines show the capturing using billing codes. So if you look across the blood components and across the sites, you see that ISBT codes captured the blood transfusion, the blood components transfusion, much more than billing codes do. And also the capturing of these components is more stable over time. We are looking at the data from 2010 to 2017. Now, one note that I should make here is that the use of ISBT codes became standard and more common as of 2012, 2013. So if you look at the data here, you see the stability of data a little more after 2012, 2013. We also selected a transfusion-related adverse event, which is trolley transfusion-related acute lung injury, as a test case to work on some outcomes. And what we did not intend to focus on, but we ended up doing, was looking at the temporality, establishing the temporality between the blood transfusion and the development or diagnosis of the adverse event. From claims data, we had an understanding that this was a challenging task to accomplish, but we thought that having access to EHR data would make this task a little easier. So I'm trying to express this challenging task on this diagram with some data. So each line here represents a patient, and we have patient IDs here. So we have put each patient on a timeline for a specific hospitalization. We have, this is the 0 line. The time after 0 shows the time after the trolley diagnosis was made, and the time below 0 is the time before the trolley was diagnosed. And the trolley here is represented with a star, and the colored squares show different blood components transfusion. So we also have the date of admission to the hospital and date of discharge from the hospital. So For a case such as patient number 4 or patient number 13, it is not too difficult to establish the temporality between blood transfusion and development of the trolley. You see that the patient is admitted to the hospital, has some transfusions, different components, and then develops a trolley. But when you look at a case such as number 59, the very last line, then this picture is not very clear. The patient is admitted and discharged from the hospital on the same date. Also on the same date, the patient receives the trolley diagnosis, but all of the blood transfusions are occurring after the diagnosis of trolley. So cases like this are numerous within the data that we work, and this is still a challenge for us to resolve. So this is just one challenge that I mentioned, but I would like to share with you as a community some of the lessons that we learned from this past year. And particularly, we were very excited about working with EHR data. And we thought that it would resolve many, or it would answer many questions. But as you know, each system brings with it its weaknesses and its strengths. So first of all, just having access to large-scale databases, even if they are EHR, does not provide the capability to run an active surveillance program. So we realized that we needed to build a fair amount of infrastructure. Then the issue of data quality was something that we had to deal with. So because we had data sources from diverse sources, the quality that each of these data sources would bring to the table was different. And our task was to standardize and harmonize these databases to a certain level acceptable to the activities of FDA to be able to use them. Even with having a common data model, we realized that the process of mapping from the source data to the common data model was not exactly standardized. There were still different rules that were applied to this process at different sites. Also, stability, consistency, and completeness of data is very important. So the bottom line is that the type of data and the quality of data that would be useful for FDA purposes is perhaps somewhat different from those conditions in a research environment. And FDA has a very high bar for data quality because the kind of activities that we do would have regulatory implications. So the 2 one-year programs will end at the end of this month, and they have proven to be successful. So CBER decided to extend the BEST initiative, and through a competitive process, we have awarded 2 new contracts, similar to the last year's 2 contracts, to several organizations to continue this work with us. Again, contract 1 would focus on surveillance infrastructure, and contract 2 would focus on innovative methods. Now the organizations that we are working with include IQVIA and OHDSI working together. We also have Acumen, IBM, and Dovell Technologies. I thought I would also share with you some of the big picture challenges that we came across in our work, particularly last year, but in general from our experience. The issue of data quality, I think, remains to be solved, and perhaps developing some standard data quality control processes and measures would not be a bad idea for the community to consider. Even setting up a threshold for an acceptable level of data quality. Also, perhaps coming up with best practices and some curation methods that everybody could follow if we are all working with the same common data model. I mentioned the temporality issue between events, which does not remain just with blood transfusion, but with almost everything, any kind of assessment that we do. And the next step, I think, in our area of work, to be able to take a couple of large steps would be to enrich our databases by linking claims and EHR data sources. I'd like to acknowledge some colleagues and collaborators. Thank you.
Okay, thanks very much for the opportunity to present. As Azadeh mentioned, I'm going to describe BEST Program 2, and we're spared going through the background information again because the scientific collaborators and the data resources, the electronic health record data resources, are exactly the same as for Contract 1. The title is Development of New and Innovative Methods for Automated Reporting for CBER-Regulated Biologic Products. And what I wanted to start with is a question that did come up in the course of the contract. Why is FDA interested in this, and how does it fit into the regulatory paradigm? So shown here on this slide is just a typical product life cycle for a regulated drug or biologic in the FDA. Of course, on the left, it has a clinical investigation plan and the different phases of an investigational new drug study. Submission of data for biologics license approval to support approval for marketing of a biologic. And then the next stage, once it's marketed, is what's known as phase 4 and post-market studies. These studies can either be passive surveillance, which is both manufacturers and consumers submitting reports to the FDA when there are adverse events, through the FAERS and VAERS system and the MedWatch system. And then active surveillance, which is what Sentinel does, which is actually going out and using real-world data and capturing adverse events and that experience from a different aspect. The major program goals of program number 2, first of all, is to develop an infrastructure to improve the quality, i.e. the accuracy and predictive value of active post-market surveillance beyond what is available solely from the claims-based big data resources that are based on billing codes. And this goes back to the initial requirement from Congress as part of the Food and Drug and Cosmetic Act amendments of 2007, not 2207, sorry. And the goal is to improve the quality of not only outcomes related to drug exposures, but also the exposures themselves, and try to improve the quality and granularity of the exposure measurement. And as mentioned, the first year really focused on blood transfusion. A term known as hemovigilance is adverse events related to blood transfusion, because they had some specific unique characteristics and unique challenges for collecting post-market data. I did want to mention as a footnote that blood products are an exception to the standard life cycle. Pre-market review is based on the adequacy of manufacturing procedures, not efficacy, because blood products have been around, blood components have been around for a long time, and their efficacy is well established. The second major goal is to increase the efficiency of collection of active post-market data, i.e. reduce the burden. For many FDA-regulated products, reporting is both electronic and required to FDA for serious adverse events through the MedWatch, FAERS, and vaccine reporting systems. Many thousands of reports are received through these systems, and they tend to be of variable quality. And there's actually some artificial intelligence programs being developed to help triage these reports to make them more amenable to medical officer review. Again, unique to blood transfusion, post-market reporting is not required at this time. There are mechanisms for voluntary reporting to FDA, but these are essentially non-existent. You might get 20 to 30 reports in a given year. U.S. hemovigilance overall, both public and private, there have been many attempts, but it's been very challenging, in part because there's no national healthcare system, poor data interoperability between hospital systems, and the complexity and limited resources of the hospitals that would need to do the reporting. In addition, adverse events related to blood transfusion, fortunately, are rare, and they're complex diagnostically because many of the things that happen in relation to a blood transfusion could also be related to other things that happen during a hospitalization. Much of the work in this program is development of what's known as computable phenotypes. And this is a phenotype that reflects the description of an individual that's a member of a cohort deemed to have an adverse event. At the simplest level in the top, you can use outcome and diagnostic codes alone, such as one might have from claims data and EHR data to define an exposure and an outcome. Moving a little further, you can use constellations of codes, such as enhanced claims data that might contain MADRS, NOMAD, other codes that reflect a medical condition, and start to build a picture of an adverse event. Taking it a step further, you can build a computable phenotype, which uses available structured data as well as data mined from clinical and nursing notes, radiology reports, lab values, and so forth, to build a computable phenotype, which then becomes a data point in its own. And then really still the gold standard for this process and used for the evaluation of the quality of computable phenotypes is case validation by clinical review of charts, which may be adjudicated or not, but increasingly is becoming semi-automated and therefore a little more amenable to large-scale studies. So the first work stream of 5 that was undertaken in this program was to improve the sensitivity and granularity of the transfusion exposures compared to claims data alone. As Azadeh mentioned, a major step forward was the ability to import ISBT 128 codes and identify products with accuracy, but still more granularity was needed to define the time of transfusion, the starting time, the stop time, events that might occur during the transfusion event. So work was done between the Georgia Tech group and Columbia University to take advantage of the fact that a standard of care is that any transfusion event is accompanied by nursing staff who watch the transfusion and make notes during the administration of the component. So the team used these documented transfusion events to use natural language processing between Georgia Tech and Columbia to mine data from these nursing notes and with a high degree of accuracy, look at transfusion start and stop times, the product definitions, vital signs in the patient during the time of transfusion, and any reactions that might occur. And ultimately, we're able to look at 34,000 transfusion records, which, believe it or not, 100% accuracy against, I think, a validation group of 100 that were looked at. So really very strong ability to insert granularity into the exposures. The second work stream is the first of 2 outcome measures that were evaluated, and this is in relation to a transfusion outcome known as post-transfusion associated circulatory overload, or TACO, and simply too much volume following a transfusion. The difficulty here is it's complex because many things in medical practice can result in circulatory overload. So one needs to be able to tie it to the transfusion event and try to evaluate it based on the signs and symptoms that are there, as well as the diuretic drugs that are provided, and do it all in the time frame of a certain number of hours following transfusion. This work was done at Stanford University, and what was found ultimately that building an NLP model by itself was not highly predictive, but using both NLP and machine learning, which was educated by known TACO events, that one could actually build a combination of code and NLP model that had a predictive value that was 16% greater than the codes alone. So this is, I think, an advance in really what we were looking for to improve the quality of outcome definition. And then the second post-transfusion outcome that was looked at was through Regenstrief Institute. Again, another difficult one to look at, post-transfusion bacterial infection or sepsis. This is difficult in part because sepsis is not uncommon in a hospital environment due to nosocomial exposures, and often the signs and symptoms can be overlap with other medical events. It's also very rare post-transfusion, about 1 in 100,000. So it's difficult to detect these out of a large number of cases. The work that was done through machine learning and NLP at Regenstrief was to look for fever over 38 degrees centigrade with a 1 degree rise in temperature following the transfusion event. And this work is still ongoing, but validated cases were identified despite the rarity of the event. And I think that work has a good start and will continue from the findings of this program. Work stream number 4, it was quickly realized that although much of the NLP development work, the foundations were built with Jon's group at Georgia Tech, upon transfer to the data partners, they were often reloaded into existing NLP platforms, and this took a fair amount of time and effort. So recognizing that commonality in an NLP platform would be very desirable. The Clarity NLP platform from Georgia Tech was offered to the data partners and was fully implemented in Columbia, and the work described from Columbia was all done on the Clarity NLP platform, which performed quite well. And then finally, the last part of the program was the 5th work stream, which was done by Georgia Tech, and it's infrastructure building to support scale up of the basic idea of what we're trying to accomplish in terms of automated adverse event identification and reporting. And this diagram here really captures it in a nutshell. If you see on the upper right-hand side, let's start in the center. In the center is an adverse event surveillance OHDSI platform. This carries the name ESUP. For those of us who actually managed to survive childhood without a computer, Aesop's Fables was probably part of our childhood. I know it was with mine, so I love the name. But this platform, shown here in the center, is capable of conducting queries both from a database that's coded to OMOP, Common Data Model, through the Atlas utility, as well as ultimately through a FHIR server that is still, in a lower maturity level, but would ultimately be found probably in most US EHRs in the next couple of years. So ESUP will conduct queries from one of these two options, bring it in, eliminate duplications, and process it through the FHIR clinical query language into an ICSR submission that can be electronically submitted to the FAERS gateway at FDA. And this was actually done. The synthetic case was built through the MIMIC database, run through this process, and prepared for electronic submission and reviewed by FDA as being eligible for electronic submission. So I think this is really a major development, not only a new tool for the OHDSI group, but also I think a path forward for automated submission of this whole program once some of these computable phenotypes are developed. So future challenges for this innovative work, timestamps. Being able to show time relationships derived from EHR timestamps would be a huge advantage to trying to describe events. Iterative computable phenotype development processes are of high value, but can we improve the efficiency in some way? These are 2 AEs for a set of products, and we ultimately hope to do all of CBER, which is many, many AEs. For the firework, collaboration with the EHR vendors and with HL7 will be needed to support eventual scale-up, but I think we're certainly on the right pathway. So shown here is a list of collaborators. I'm sorry it's a little bit small. You might not be able to see it all. I just want to say on both behalf of OHDSI and myself and our staffs at FDA, it was really a privilege to work on literally a weekly basis with the OHDSI principals on this program. Really a lot of scientific horsepower went into this, and we appreciate it. And I specifically want to call out Jon Duke and his group, which Jon was, I think, the unofficial captain of the team, produced a lot of the fundamental foundational science that went into it, and held us to a high standard for quality and productivity. So thanks, Jon.
Well, thank you both to Azadeh and Alan so much for that. I will say if there's one burning question, can we get time in for one or two questions for Alan or Azadeh, please? Okay, everyone knows where to find us afterwards for follow-up. We appreciate, again, so much the opportunity, both for the collaboration and for their time today. So thank you, Alan and Azadeh.